Hello, I'm Andrew Pryor, and my project concerns a self-propagating kill switch signal. As genetically modified organisms are engineered for new and diverse purposes, it becomes critical to develop safeguards that can regulate the spread of engineered organisms to prevent the contamination of natural ecosystems. These safeguards are especially relevant where genetically modified organisms are deployed on large scales and in open environments for purposes such as bioremediation, farming, and biofuel production. For purposes such as these, demonstration of fine control over engineered organisms become necessary to mitigate fears of environmental damage and persuade both the public and government regulators to more readily accept genetically modified organisms. To this end, genetically modified barriers or safeguards such as biological kill switches, trophic dependencies, or semantic containment have been developed. This work proposes a new system in which a biological kill switch can be coupled with a self-propagating signal. This system has two independent functions: a non-propagating kill switch signal that is limited to only cells receiving small molecules, or to trigger a kill switch signal via small molecules that will then self-propagate through the population. This circuit describes the cascade of events inside the cell to achieve these behaviors. Addition of only small molecule 2 results in a cell death that is localized to the cells only receiving the inducer. Addition of small molecules 1 and 2 together results in a spike of AHL followed by a delayed accumulation of toxin. The spike of AHL enables the kill switch signal to be passed to surrounding cells. Neighboring cells will receive the AH signal, triggering a cascade that will rapidly produce more AHL, then cause cell death. Antitoxins are also included to prevent natural fluctuations from triggering the kill switch. The benefit of the self-propagating signal is that you do not need to rely on added inducer to reach every single cell to destroy the entire population. My project has 13 compositors with 13 parts. It will explore the necessary parameters to achieve this model, even though the parameter space is very large. We'll only look at a small subset of these. I first began testing a basic system for the interaction between the four proteins controlling the expression of AHL, toxin, and antitoxin. The graphs on the left show the first test with small molecules in 2. This test uses parameters of protein A, B, C, and D interactions for strong binding, slow decay, and fast generation. It can be seen here that the fast production of antitoxin 2 leads to such large accumulation of antitoxin 2 that toxin 2 is unable to accumulate to lethal levels despite repression of generation of antitoxin 2 by small molecule 2 inducer. This result led me to identify needed adjustments to the model. In order to achieve desired function, the KD value of antitoxin 2 could be increased, the production of antitoxin 2 could be increased, or the production of antitoxin 2 could be decreased. The graphs on the right show the result of parameter adjustments. It can be seen that a spike of small molecule inducer 2 quickly leads to a spike in toxin 2, causing cell death. The KD value of antitoxin 2 has been increased to KD equals 5. The production of toxin 2 has been increased to Qtox equals 0.4 by increasing the KD value and the decay rate of protein D, and the decay rate of antitoxin 2 has increased to 0.2. The production of antitoxin 2 has also increased to prevent generation of a hazard caused by toxin 2. I next tested the model for both small molecule 1 and 2. This result showed a system failure in which AAL was not triggered. This result was due to production terms in AHL compositor could not overcome the decay or diffusion terms. Furthermore, there was a lethal buildup of toxin 2. These results led me to identify several adjustments to improve the model. To adjust for this, the KD value and decay rates of C was increased to enable faster production of AHL. Furthermore, the production of AHL from LuxI was increased to allow for increased production of AHL and a more rapid response. As the graphs on the right show, these adjustments worked. The proper spike of AHL is seen as a result of both inducers, followed by lethal accumulation of toxin 1. The decay rate of protein C was increased to 0.008, the KD of protein C was increased to 10, and the production of AHL was increased to 0.8. These resulting changes significantly improved the AHL response, where AHL spiked to concentrations of 50 nanomolar immediately after the addition of the inducers. There was a delayed increase of toxin 1 after 300 seconds after the spike of AHL as designed. The second copy of antitoxin 2, which is promoted by SM1, has successfully repressed the spike of toxin 2 when triggering population cell death using both SM1 and SM2. In this slide, the analysis is shown for 0.001 nanomolar of AHL added to the system at time equals 500 seconds. The results show a strong response after a small delay of approximately 60 seconds. AHL then accumulates to reach a concentration of 35 nanomolar. Approximately 300 seconds after the spike in AHL, toxic 1 accumulates to lethal levels. This result shows the sensitivity of the system. In fact, by further decreasing AHL inducer, the lower limit of the inducer was determined to be approximately 0.004 nanomolar. Using the model developed by Gelini in 2012, this range of concentration from the initial spike of AHL to the diluted concentration correlates to a spatial distance of approximately 1.5 centimeters, allowing microscopic influences from one side of the solution to the other. This model proves the circuit functions within physiological limitations, has a fast response time within less than 5 minutes, and has a high sensitivity of 0.001 nanomolar or 0.004 nanomolar at the limit. This means the kill switch can effectively and quickly kill a bacterial population and enable precise control over such a population. Further testing is required to determine the stability of the system in vivo as well as the metabolic load placed on the cell by the system.